Well, thank you for joining me on this exciting morning. I've turned my video off just to avoid any network issues while we discuss this work. So today I'll be talking about our work on using imaging, in particular cell painting, and machine learning for problems in drug discovery, and we'll learn more about that as we go through our discussion today. So our group focuses on microscopy images because there's just so much information present even in a single image. And I know I'm preaching to the choir here, but with modern image analysis software, it's possible to measure most phenotypes in most biological systems of interest, including complex culture tissues, culture systems, patient tissues, and so on. And we can do this for both time-lapse images as well as for 3D. But what does this technology equip us with? The most obvious application is to measure specific phenotypes in images, which requires, of course, that you know the phenotype you're looking for ahead of time. I'll just walk you through a few examples of this application. So this example relates to cancer. Our collaborators were looking for compounds that could transform leukemic cells into harmless polyploid state, where the nuclei have more than 2 pairs of chromosomes, shown here on the right, where the software generated outlines. We created an image analysis pipeline to detect this transformed state. You can see the difference in nuclear size between the red and blue, corresponding to control treatment and a possible drug for leukemia. We obtained several hits in the screen. We tested them on human patient blood samples, which confirmed that a drug they identified is able to reduce the leukemic content in those isolated samples. This led to a clinical trial starting a few years ago for alisertib in adults with this form of leukemia, and it was reported earlier last year that results looked really promising. So that was an approach where we knew exactly the features to measure, but we do extract hundreds of features, and sometimes it makes sense to take a machine learning approach to identify phenotypes of interest. If we don't already know the exact feature we care about, but we do have positive and negative examples of what we are looking for. So here's an example of that. Our collaborators assessed which compounds may restrict the growth of leukemic stem cells. Targeting these cells is crucial, but it's a difficult goal because normal and cancer stem cells look quite similar. But our collaborators observed that leukemic stem cells tend to have a cobblestone-like appearance, so when they're grown in a dish, which is shown here in large clumps. So now the goal became to come up with a way to reliably detect these clumps of cells and then screen for compounds that result in a reduction of these clumps. We used various image-derived measurements to train a machine learning algorithm, a classifier, to detect this cobblestone appearance, and then used it to screen about 14,000 compounds. We identified compounds including an FDA-approved drug for a different indication that could selectively kill leukemic cells. So those were 2 examples, but we've used image-based screening in many projects, and I'll just display a range of applications we've worked on. We've used images for counting hepatocytes, counting speckles of DNA damage, scoring failed mitoses, measuring mitochondrial abundance, counting worms killed by pathogens, measuring how fat C. elegans are, measuring TAR pathway activation, meiosis markers, metastasis, and others like neurofibrous structure in glioblastoma to infection by liver stage malaria, and many more. And we've also done this in multidimensional images, including time-lapse and 3D images. And all this work is having an impact. These are just some of the cases where compounds discovered using our team software and methods have been shown to be effective in animal models of disease. As I mentioned, one clinical trial has already begun for myelofibrosis, which is already showing clinical benefit, and others are moving in that direction. All of this work has led to advancements to CellProfiler, which is our open source software for image analysis. It's widely used around the world, not just in academia, but also in pharma companies, and you can check it out at cellprofiler.org. We're also working on the next generation of computational imaging techniques, in specific leveraging the rapid developments that have been made in computer vision and deep learning. Computer vision, as Anna will talk about probably later today, is a field that's focused on analyzing natural images like faces and scenes. And by about 2012, modern artificial neural networks has beaten the state of the art on nearly every major computer vision problem. For example, the problem of finding faces in a picture can be done very easily now using neural networks. As it turns out, the same neural network models that are used to find faces can also be used to find nuclei. And all you need to do is to provide examples of outline nuclei and then train the model to find those outlines. So given this advancement, we hypothesized that a single robust model focused on a specific problem, like identifying nuclei, can remove the need for tweaking pipelines in each experiment, like we currently need to do in CellProfiler. This was the motivation for our organizing the 2018 Data Science Bowl. For this challenge, we created a large data set with images from multiple experiments and hand-annotated thousands of nuclei. We asked the data science community to identify or segment all nuclei in a test data set as accurately as possible. About 4,000 teams from all over the world created machine learning models to solve the nucleus segmentation problem in a wide context. Any microscopy images acquired in nearly any modality, and we found that the top performing models were able to outperform in this wide context solutions by image analysts, and that suggests that in the near future we may be able to develop fully automated image analysis solutions for some specific yet crucial problems such as identifying nuclei. We are revisiting the phenotype classification problem as well. That is how to automatically sort cells into phenotype classes, and are seeing that neural networks can beat state-of-the-art algorithms in identifying cellular phenotypes. Peter Harwood's group and ours decided to combine efforts to replace our existing tools for this purpose, CellProfiler Analyst and Advanced Cell Classifier, to come up with a new deep learning-based tool for sorting cells based on phenotypes, and then training classifiers to automatically score millions of cells on these phenotypes. So that's the aspect of imaging where we measure specific phenotypes that one cares about. But you can use images and image-derived measurements in a more unbiased way, which is to profile samples and to create signatures of cell state. So several of you may already be witness to a shift that's been taking place in our thinking about images. That's the information that we extract from images can be in fact as computable as genomic information and many other such large information sources that we have in biology. So the hypothesis here is that what if images contained enough information to characterize a reasonably complete picture of cell state? Now, we know that at some level this is true. In classic genetic screens, seeing the same phenotype means that genes in the same pathway were perturbed. So the question is, can we just take this basic concept and scale it up? We can first scale it up by staining cells with as many dyes as we can fit into a single assay. We ended up with 6 different dyes, which label 8 different cell constituents that are imaged in 5 channels, shown here. That's the cell painting assay, which we developed with the Schreiber lab at the Broad. It's not a complicated assay. These are cheap dyes. So this is a very high throughput, inexpensive, non-fussy assay. You can run it yourself using a regular lab microscope, using this 2016 protocols paper as the guide. And then we can also scale it up in terms of the number of perturbations we can measure with automated microscopes, and then scale it up again by extracting thousands of features and metrics from each cell. And the question then is, will there be changes in the quantitative metrics that we cannot discern by eye that nevertheless indicate similarities in the underlying cell state? Each perturbation we test, that's a chemical or genetic treatment of some kind, is therefore turned into this rich profile of single cell data. And then by measuring similarities between profiles, we can dramatically impact different stages of the drug discovery pipeline. You might have already gotten a sense of how powerful profiling can be to this end, but I'll just give you a sense of that through some examples of applying image-based profiling to drug discovery. Before I get into examples, I just wanted to point out 2 things. First, that image-based profiling assays indeed produce hundreds of measurements per cell. We extract simple features such as counts, sizes, and intensities, but also more complex ones that are inherently multiparametric, such as shape and texture. We also measure correlations between channels and neighborhood relationships between cells. And once we do this across multiple channels and cell contexts, once you count it all that, we end up with hundreds of measurements for each cell. And then second, one may wonder whether there is indeed that much information in an image to be able to tell what cell pathways are perturbed and so on. And here's just a simple example of getting an intuition about that. Here are some images of cells as they look under a microscope with no labels or stains and no fluorescent imaging. And I bet it's hard for you to figure out, what stage of the cell cycle each one is in, except maybe the telophase to the right. But machine learning models trained on this problem can very easily classify these stages. And I might be, again, preaching to the choir here, because as you see in this confusion matrix where the actual phases are shown in rows and the predicted ones are in columns, you see that most of the predictions are correct, following along the diagonal with only a few mistakes on the off diagonal. And this model is based on dimensions features on thousands of dimensions, but you can also view single cells in this simplified three-dimensional version, this place, and see how cells segregate in space based on their cell cycle stage. So this is a good demonstration that images indeed have much more information than one might think, and certainly much more than what you can see by eye. So let's apply that information and images to important challenges in drug discovery. This is an example that you might have seen many times before. It's one of the earliest applications, which was grouping chemicals based on the morphologies that they induce in cells and assigning mechanism just by guilt by association. That is, compounds that induce similar phenotypic response may have this similar mechanism. And as you can see in this experiment, this approach worked. Compounds that are already known to have the same type of activity grouped together. Most predictions correctly fall along the diagonal. But this experiment was a simple case of using strong compounds with visibly obvious phenotypes, so it was far too easy in that sense. But more recently, the connectivity map team at the Broad ran a pilot study using more than 1,000 compounds from a much more diverse set of classes of activity. And here, given that many compounds mechanisms are unknown or even overlapping, we do not expect to see a strong diagonal. So we've ordered the rows and columns to put the categories of compounds with the strongest signal to the top left. And we see a decent proportion of categories yield a signal along the diagonal, and also that some categories show similarities to other categories. So a really great thing about this experiment is that we also have gene expression data from a high throughput transcriptional assay of 1,000. So you can see how that behaves on this task here. And when we look at which categories are well captured, we see some shared strengths between the two assay types, but also a lot of differences. That is, we see a lot of complementarity. So what we're really excited about is the prospect of taking both kinds of data, making joint profiles and increasing the power of the profiling approach. Aside from compounds, we've also tested genetic perturbations and found a similarly impressive ability to cluster them based on similarity, which can tell you about gene function. When you overexpress each gene in a cell population, then cell paint them and extract profiles, you can successfully rediscover in a single experiment all these known biological pathways. For example, the RAS pathway up here, and the Hippo pathway here. And we also see sensible anti-correlations, for example, negative versus positive regulators of the same pathway. In fact, we discovered that an unexpected such relationship between the Hippo and NF-kappa B pathways, which we later confirmed in follow-up work. And beyond looking at gene function, we might also be able to stratify patients and characterize disease alleles based on morphological profiling. And here's an example in lung cancer. So like for most diseases, it's not a single disease, but can instead be caused by a variety of underlying mutations. But we can overexpress each disease-associated allele and look for similarities among their profiles. And we might discover that you're not dealing with 353 different types of cancer, but instead a small number of subtypes. The grouping of genes here broadly fits known biology, but more interestingly, looking within a given gene, for example, BRAF here, we find that the similarities we find between alleles mirrors what is known about their activity. For example, the type group up here are known to fall into a similar class. We overexpress the wild type here at the bottom, and we overexpress each mutant, and we see which ones yield similar morphological profiles. There's an exciting technology underway that could really scale up these experiments. The Blaney lab at the Broad has developed a technology to do pooled image-based screening, and we are now working with them and the Neal lab to create a pooled version of cell painting. The idea is to perturb cells using a pooled perturbation library, for example, a pooled CRISPR library, and then do cell painting to get per cell image-based profiles, and then also read out the barcode of the guide RNA present in each cell. The trick is to first generate many copies of the barcode in situ, then sequence them in situ by imaging using sequencing. That means you can assign perturbations to each cell, as well as have the image-based profile of each cell. This can be a game changer in terms of massively scaling up cell painting genetic screens, like the one I just described on the prior slide. And in fact, during the pandemic, we did our first whole genome screen, which is still being analyzed, but the the excitement is really up there, because it would have been impossible to do this at the same scale in an arrayed format. The end goal of all these efforts is to discover novel therapeutics, and I'll show you just two stories about how image-based profiling might do this. The following is a relatively straightforward approach when it's known that a particular mutation is causative for a disorder. A team from the University of Utah showed that for a monogenic loss of function disorder like CCM, you make the loss of function in cultured cells and see if there's a detectable difference. Here, the difference is obvious. You don't need fancy image analysis, but whether it's obvious or subtle, once you have found a phenotype, you can then screen for drugs that revert that phenotype back to a healthy state, back to a healthy phenotype. And the academic team here then launched a company based on this work, and has now drug screened more than 200 rare disease models using cell painting. So that's highly parallel drug discovery that requires very little customization, or almost no customization, to each disorder. And this is, in fact, an idea we are pursuing in our collaboration with Mikko Taipale and University of Toronto in the context of rare diseases. There are over 7,000 diseases categorized as rare, of which 4,500 are known monogenic. So only about 6% of these have FDA-approved treatments, and the path forward is not clear. The traditional approach of using multiple assays and technologies to study each disease will be impractical, but this could be flipped around. What if we instead profile thousands of diseases with a few broadly useful assays to gain insights into their pathology? Image-based assays can be a rapid, relatively low-cost, yet information-rich approach to achieve this goal. The Taipale lab has created an image-based profiling data set for more than 1,000 Mendelian diseases across more than 3,500 mutant alleles by overexpressing them in a cell line. Their initial goal was to sort of systematically characterize the protein localization of disease variant by scoring each of these images by eye. There's a grad student who's sort of doing this for 3 months at a stretch. And we're now taking a fully automated approach, taking an unbiased approach to this problem that is looking not just at protein localization, but a broad set of morphological measurements to find unique signatures of mutants. For mutants with distinct profiles, we could then get some insight into what are the structural changes in the cell, but then also be able to screen for drugs that can revert that phenotype. In fact, we've already leapfrogged to actually doing the drug screen using the pool cell painting technology I just described, because that allows us to test out hundreds of alleles against a drug in a single well. There are many other applications of image-based profiling that we've been pursuing, led by our collaborators. I'll just walk you through two of these. So large-scale assays for drug discovery can be expensive and impractical, particularly for model systems that require primary cells, organoids, or whole organisms. But as an alternative, an international team, including Janssen and many others, demonstrated that data from a single high-throughput imaging assay can be repurposed to predict the biological activity of compounds in other assays, even those examining unrelated pathways or biological processes. This proof of concept results suggest that image data from high-content cellular assays can be leveraged to carefully design chemical compound screens and boost hit rates. And we have sort of been repeating this whole concept, but sort of scaling it up at the Broad in terms of the number of different data sources we integrate. We're now integrating gene expression data and chemical structure data, and coming up with predictions of these assays using a similar idea, but using cell painting as the input source. And second, in this project, we are using cell painting to come up with pharmacodynamic markers of resistance and sensitivity. We take a set of paired cell lines where one of each pair has been modified to render resistance to a specific drug. And we then profile the cell lines before and after drug treatment. Then we investigate whether the drug treatment, the drug resistant or drug sensitive cell lines have any morphological traits in common. That is, are there morphological markers of drug sensitivity or resistance? And if we find such markers, this could be useful in determining drug susceptibility for individual patients' tumor cells. I just wanted to briefly discuss two ideas we've been pursuing to increase the interpretability of image-based profiles. As I mentioned earlier, we're extracting hundreds of measurements per cell, and many of these measurements do not have obvious biological meaning. This problem is even more acute with deep learning-based feature extraction, where it's nearly impossible to attribute meaning to individual features. But one approach to increase interpretability is to predict other readouts that are more interpretable. Piquito Vasquez and others at the Broad had created a cell health assay that reads out a set of 70 or so phenotypes, including proportion of cells in specific cell cycle stages, the kind of apoptosis initiated, cell proliferation rates, reactive oxygen species present, and so on, which are much more biologically interpretable. We collected genetic perturbation data across 3 cell lines for a small handful of genes in both the cell painting assay and the cell health assay. And then we used the cell painting features to predict these cell health phenotypes. Greg Way, a postdoc in the group, has shown that we can do a really good job of predicting several of these readouts, and you can learn more about this project on GitHub at this link, and the preprint has also recently come out with all the details of this paper. In this line of work, for interpretation, we take a visualization approach to gain insight. We generate synthetic images of cells to visualize the structural changes a cell undergoes when treated with a perturbation. So given several examples of cells treated with compounds, the question is, can we learn a model that can not just synthesize realistic images, like you see in this animation, work by Nick Pawlowski and Peter Goldsborough, but also allow us to interpolate between 2 endpoints. For example, if you take a control treated cell on the left and a cell treated with a compound on the right, can we interpolate between these 2 points to reveal the gradual changes that the cell may have undergone? Generative adversarial networks, or GANs, are a class of neural network architectures that allow you to do exactly this. All these images in between were generated by Maxime Lafarge by interpolating between 2 endpoints in a high dimensional space corresponding to control and the compound, revealing the gradual changes in image phenotype. We asked expert cell biologists, including Anne, to score synthetic images to be real or fake, and we found that the network could fool them about 40% of the time. So what's really great about this is that you can use the same representation that gives you the synthetic images can also be used as profiles for the perturbation, and that in turn can help make the profiles more interpretable. In closing, I just wanted to mention the joint undertaking in morphological profiling, or JUMP, using cell painting. It's a consortium of about 10 pharma companies and 3 academic groups led by us at the Broad, who are committed to creating the first large publicly available cell painting data set. This data set will generate over 140,000 compound profiles and over 10,000 genetic profiles, which will be done across multiple sites. And through this data generation process, we aim to address several assay development and computational challenges in cell painting that could further accelerate its use in drug discovery. And we're soon coming out with a newsletter that'll help you keep posted about our progress on this project. So that's about all I wanted to discuss today. If any of this interests you, do reach out, but also consider attending CytoData 2020, which is just a month from now, and it's virtual. It's a symposium that's 5th year and running, which focuses on problems in image-based profiling. And I'm super excited that Recursion Pharma will be the ones organizing it this year. So with that, I would like to thank everyone who's contributed to these exciting projects, and I am happy to take questions.
That's great. And so I'll do the virtual class, Shanantu.
Thank you.
That was fabulous. Thanks so much. So there are questions lining up in the chat, which is fabulous. I'll give people a couple seconds to type a few things. Maybe I'll just ask a first question. Shanantu, do you have enough experience with the various features that you're collecting now to know which you actually need to work at the cell level to get some sense of heterogeneity versus which you can actually do some kind of simple median or whatever, just to reduce the computational load? So which of the features, I guess, which of the features are really signaling heterogeneity in the populations that you're looking at?
Wow, that is an interesting question. We actually haven't narrowed it down to we haven't studied this in any systematic sense. We've mostly taken a broad signature-based approach to it, and sort of directly tried to model the heterogeneity by either finding, in this large phenotypic space, the clustering of single cells, and find whether there are cases where the differences between 2 conditions is actually the differences in the demographics of the 2 cells, of the 2 set of the two populations, the relative proportions. We actually never narrowed it down to a small set of features. We've also sort of taken a more sort of statistical approach where we've tried to model the covariance structure of the data and so on. So in all those cases, we're looking at multi-parametric readouts. I mean, all readouts together never really narrowed it down to specific categories. But that's a great question.
All right, thank you. Okay, so I will invite people who've put their questions in the chat to turn on their video. Yes, first up is Sayan Biswas. And Sayan, can you turn on your video, obviously your microphone, and also tell us where you're from in the great spirit of global bioimaging. I don't know where he is. Don't have. Sayan, we don't have your video as far as I can tell. Or if you want to just turn on your audio. So, I'll tell you what, while Sayan's getting ready, why don't we move to Claire Brown from Canada. Claire, do you want to turn on your video and ask your question?
Yeah, so it's Claire Brown from McGill University in Montreal. And thank you for the great presentation. I'm really fascinated by this field and the fact that you can generate this synthetic data, but are there efforts to protect against fraud where it's so easy to generate these type of realistic looking synthetic images now?
Wow, that's an excellent question. I wish I was able to quickly pull up the paper I actually came across very recently. So by the way, none of it is, we haven't made any efforts on that front ourselves, but just a few months ago, and I'll try to post the link in this chat window once we're done, I came across this work where someone had taken the Broad Bioimaging benchmark collection that we've curated at the Broad and used that to do exactly this, which is to sort of get positive examples of classes of sort of bioimaging data. By positive here, I mean real, and then use that to develop a network to detect fakes. And indeed, there is some work in that space. I'll try to pull it up.
Great, thank you.
Yep.
Okay, we'll do one more. Has there been any effort to move to in vivo analysis? Oh, I'm sorry. Andrea, can you turn on your camera? It is 5 a.m. here. Okay, so maybe not. So why don't we just address Shanantu Andrea's question? Any effort to move into, for example, zebrafish, et cetera?
Right. So no, well, we do have worm imaging, and I'm sorry, I'll have to quickly run through my my repository of memory on this front. So we do have ongoing efforts with worms. I believe there are projects with zebrafish, but I don't think those are in vivo at our end. There are groups at Harvard, Sean Megason's lab at Harvard. They developed the goat figure tool a few years ago. So there are certainly ongoing efforts there in Boston, but not ourselves, as far as I know.
Okay. Let's see. One last question, and then we'll move on. And I encourage you to keep typing questions. And Shanantu, I don't know what your schedule is, but if you can hang around for a little while, address questions while they come in.
Sure. As soon as you hear a toddler, that's my time up.
Okay. All right. Nicole Salgado from Chile. Nicole, if you're there.
Yes. Hi, everyone. Thank you for your talk. It was very, very interesting. I was wondering if there is any repository of these synthetic images to simulate other type of analysis.
A repository of synthetic images specifically, or of real images to generate synthetic images?
Both.
So
because
of real images. Go ahead.
Yeah, I was wondering because sometimes here we don't have certain technologies. For example, there is no microscopes here to perform FLIM, and therefore images to recreate this kind of analysis could be very useful to develop new technologies for analyze, even when we don't have the technology of the imaging yet?
Oh, interesting. Yeah. So there's certainly large benchmark data sets. In fact, Jason would be the right person to address that. The image data repository, the image data resource is a fantastic source of massive amounts of imaging data that would help with sort of getting you started with existing data for doing the analysis. In terms of generating synthetic data, I'm aware myself immediately of one such resource. I'll try to search for that and paste it in here. I believe this is from Peter Harwood's group, where they're able to generate realistic images of cells. I believe they'd used it for their cell segmentation exercises. So there are those sources too, but I think if you're sort of trying to bootstrap an analysis, you'd probably want to use existing data where you have some annotations or some sort of a scientific context. And the image data resource and the Global BioImaging benchmark collection are both great. I'm sure there are many others that are similar. UBIOS has been creating a resource that is similar, as far as I know.
Okay, thank you.
Thank you all. The questions are continuing to come into the chat. So Shantanu, if you're able to address those directly in the chats, that would be great. Just to remind everybody, if you can, if you don't mind, keep the conversation in the to everyone so that everyone can benefit from that. Mindful of the time, I'm going to just give Shantanu a huge final thank you for stepping in at short notice, and also just for giving an absolutely fabulous presentation of the great work ongoing at the Broad. So seriously, Shantanu, thanks a lot.
Well, thanks for the invitation, Jason. And I'm excited to see that Anne is actually able to respond to some questions here. Anne Van Dyke is Anne Carpenter. So perhaps we have an answer for a question I didn't address. Thanks again, Jason.
Okay.
Thanks again.
All right. And I'll hand over to Noto to take us into the next session.